All right, let's talk about how the kidneys are involved in pH regulation. Now, one thing, to talk about this, we got to remember a few things. One, you've learned before, the bicarbonate buffer. This is one of the major ways that our body changes pH. Remember, water can react with carbon dioxide with the enzyme carbonic anhydrase to produce protons and bicarbonate. Protons are what makes a solution more acidic. The more of this you have, the lower your pH, the more acidic you are. So, if I add CO2 to a solution, it tends to go this way and make more protons to make the solution more acidic. If I added more bicarbonate to a solution, it would tend to push it this way, which would remove protons from solution and make the solution more alkaline. So, we can adjust pH by adding CO2 or removing CO2 or adding bicarbonate or removing bicarbonate or even adding this and removing that directly. So, that's going to be one of the tools that we use. Now, before we talk about how the kidneys regulate pH, let's talk about why pH might vary. We know that pH is important because pH affects the folding of proteins, and the folding of proteins affects what they do. So our enzymes only work because they have a certain shape, and if the pH changes substantially, then the shape of the protein changes, and our enzymes don't work the way they should. Now, on the pH scale, 7.4 is considered typical for most body fluid. Remember, 7 is neutral, so 7.4 is slightly alkaline. That's what's normal for our blood and most of our extracellular fluid. And the normal range is about 7.35 to about 7.45. Anything within that is completely normal pH, but outside of that, we consider it abnormal. Now, just a little outside of that, you won't notice any significant problems, but that's where we consider it to be indicative of possibly a problem. More acidic than 7.35 we call acidosis, and more alkaline than 7.45 we call alkalosis. If you actually get down to a pH of 7, where you're actually getting to neutral or even acidic pHs in your blood, that's immediately dangerous. You can die from that. And if you get this up to about 7.7 or 8, that's dangerous levels of alkalosis. Within that, it's not that it's trivial, it's just that those levels will indicate like rapid death. Now, what would make our pH change? Now, there's a lot of misinformation about this. A whole lot of people out there talk about how, all of these things happen because you're too acidic, you need to alkalinize your body. Cancer feeds on acid and things like that. And to be honest, that's hogwash. It's not that pH isn't important. pH matters a great deal. You can see that the normal range for pH is very narrow. But that's why your body has excellent mechanisms already to regulate pH. And most of the time, it doesn't really need your help. If you like alkaline water, by all means, have some alkaline water. But don't do it because you think it's necessary to fix a health problem. Unless you have some sort of disorder that affects your pH, if you are otherwise healthy, your body really does not need much help managing pH. It's very good at it on its own. And we're going to learn some of how that works. You already know one, which is the respiratory system. Just by regulating breathing rate, you can adjust your body's pH very effectively. The kidneys can help when that's not enough or when you need a larger or longer term regulation. We're going to see how that works. But let's start by talking about what could actually change our pH. So what makes the body more acid? Sources of acid. Well, carbon dioxide. If you hold your breath and CO2 builds up in your lungs, it then builds up in your blood, and that actually does change your pH slightly. This buffer system absorbs a lot of that change, but not all. So carbon dioxide and many other metabolic wastes. You have a number of wastes in your body which are acidic. For example, ketones that come from fat metabolism are strongly acidic. Also lactic acid from anaerobic metabolism. So either of those is a source of acid. Another big one is dietary. We like acidic foods. The taste of sour or tangy is the taste of acid. So that taste that we tend to like in, say, tomatoes, pretty strongly acidic. Orange juice, quite acidic. Coffee, very acidic. A lot of these things that have that tang, they have it because they are acidic. That's not a bad thing, but it's worth noting that we seem to like the taste of acid. I once actually heard from somebody about how they were trying to justify why they were alkalinizing their body and still eating foods they liked, and they were making a claim that lemons were actually alkalinizing. They're not. Lemons are pretty strongly acidic. I don't know where that came from. Anyway, so those are some of the major sources of acid that can add acid to your body. What about sources of base? What can make your body more alkaline? Well, hyperventilation, getting rid of CO2, can do it. If I breathe more quickly and or more deeply than I need to, I'm getting rid of CO2 faster than my body makes it. Removing CO2 draws the equation this way, which removes protons from the body, and I become more alkaline. That's actually what produces the lightheadedness and tingly feelings you get in the case when you're hyperventilating. There aren't really a lot of metabolic wastes that are strongly alkaline. And generally, we don't like alkaline foods. Most foods that are actually alkaline taste bitter to us. The only foods we commonly eat that are significantly alkaline, there are some vegetables, which are somewhat alkaline. And many vegetables are kind of bitter tasting. Normally, we don't eat a lot of alkaline foods. So, really, that's one of them, hyperventilation. The other one, Tums Antacids. It is certainly possible to overdose on Tums. In fact, I guess they see that fairly often in children's hospitals because antacids are often in bright colors and they taste kind of sweet like candy. So kids get a hold of, and a lot of people don't lock them up because they don't think of them as potentially dangerous so much. But a kid gets that and they just start chowing down on Tums. And they can put so much bicarbonate into their body, that's Tums effectively are bicarbonate, that it absorbs too much acid and they end up with some level of alkalosis. Not usually a problem, but it's best avoided. Anyway, so it's a lot easier to become more acidic than to become more alkaline. There are a few other things, but so that's one of the reasons why our kidneys actually have more ways of dealing with acidosis than alkalosis. They have more ways of removing acid from the body or adding bicarbonate than the other way. So we're going to start by talking about how our kidneys can help us when we are trying to deal with acidosis, when there's too much acid in our blood. And then we'll talk about one way that they can help us deal with alkalosis. Okay, let's talk about how the kidney can help us deal with acidosis. There are two mechanisms we're going to mention. The first, the proximal tubule can take bicarbonate that's in the filtrate and move it back into the blood. We can reabsorb bicarbonate this way. That will make sure that there's bicarbonate that was in our blood that we were going to lose in our urine that we don't lose, that we bring back to our blood. And the more bicarbonate in your blood, the more you can deal with acid. So in that situation, we are going to start with a bicarbonate ion. Here in the lumen, this is the inside of the tube, the stuff that's on its way down the urinary tubule, that we want to move here. This is outside the urinary tubule where the capillaries are. So how am I going to get my bicarbonate from here to here? Well, when we reabsorbed something like sodium, what we had was sodium channels here and a sodium-potassium ATPase here. This pumped the sodium out, which drew it in there. That would be great if we had a powered bicarbonate transporter, but we don't. To the best of, I don't know of a bicarbonate ATPase. So I need to get this bicarbonate across this membrane somehow. One problem I've got here is that I actually don't have any bicarbonate transports on this apical membrane, which is a problem. Because if I don't have any transports for bicarbonate, how can I get this in here? If only there was some way to turn bicarbonate into a form that could cross cell membranes. If only there was a way I could take bicarbonate, and make it into something which can diffuse across a cell membrane, like carbon dioxide, perhaps. Yes, that would work. Carbon dioxide would work very well. What do I need to turn a bicarbonate into carbon dioxide? Well, I need a proton, and I need the enzyme carbonic anhydrase. As it turns out, PCT cells have carbonic anhydrase enzymes in their apical membranes, facing the lumen, as a membrane enzyme facing outward. So if my bicarbonate can get a proton in there, then it can turn into CO2. How can I get a proton? As it turns out, there is a transporter for that. This is something called the NHE, which stands for sodium Na proton H exchanger. The NHE is a sodium proton antiport. So it's going to take sodium coming in exchange for protons coming out. Now, where did I get the proton? Hold that thought. We'll get back to that. But now I've got everything I need. My bicarbonate and my proton can react together at the carbonic anhydrase to form CO2 and water. And CO2 can simply diffuse across cell membranes into the cell. Now it's in the cell, but it could just as easily go back out. That's not what I want. In fact, I want to get it back into the blood in the form of bicarbonate. If only there were a way to turn carbon dioxide into something that can't just leave this cell, that would be useful to me. I could react it with water in the presence of carbonic anhydrase and turn it back into a bicarbonate ion. But I would need carbonic anhydrase, which, as it turns out, I have. Inside the cell, as well as on the apical membrane, I have carbonic anhydrase enzymes. So I can take my carbon dioxide and water. Now, sometimes the question comes up, is that the same water as it was out here? And the answer is, it doesn't matter. There's water everywhere. You can always assume that there's water if you're in the body. So my carbon dioxide and water can react here to produce bicarbonate and proton. Hey, I needed a proton earlier to come back out here and pick up that bicarbonate. How handy that I just made one. Nice. Anyway, so now I'm back to my bicarbonate, and this can't just drift out of this side. Remember, we had to turn it into something else to get it in the first place. So now I need to get it out here. How exactly that happens is something that I have read several different accounts of, and so it seems to depend on who you talk to. I'm not going to hold you responsible for how the bicarbonate gets out here. The last thing I heard that seemed the most reliable is that there seems to be a sodium bicarbonate SIM port that brings the bicarbonate out, but I'm not going to hold you responsible for that. You can just say somehow it gets out here. Net result, I have taken one bicarbonate and moved it from the lumen out of the tubule back into the body where it can get picked up by capillaries. One bicarbonate reabsorbed. Yay! Kind of neat that in the process it just sort of recycles that proton. I always liked that part. Anyway, so you should understand this process. You should be able to describe this. Now, that's helpful. But of course, that's a bicarbonate that was already in my blood, and all I did was not lose it. It was in my blood, filtered out. Rather than lose it in the urine, I put it back into the blood. That's good. But I might need something more. What if my acidosis condition is such that I need more bicarbonate in my blood, not just to avoid losing some of what I have? Well, that's where the second mechanism is going to come in. The DCT can do something a little more important, which is to take carbon dioxide from the blood and send it back as a new bicarbonate, one that we didn't already have. So that's actually going to be kind of helpful for us. So that's going to be our next thing to talk about. Okay, let's talk about how the DCT can help us with acidosis. This is going to take place in the distal convoluted tubule in something called a type A intercalated cell. You may remember that when we talked about aldosterone, we talked about the principal cells. This is one of the other cells in the DCT. This is an intercalated cell type A. Now, our goal here is going to be to take a carbon dioxide from the blood and return it as a bicarbonate ion. So I want to take this and give back that. That will result in the net gain of a bicarbonate in the blood and the loss of a CO2, which should help with acidosis. In order to do that, though, I'm going to have to convert this in the cell, and that's going to require carbonic anhydrase. So inside the cell, I should have carbonic anhydrase, and these cells do have a lot of carbonic anhydrase. So my CO2 comes in. We can always assume that there's water, and becomes bicarbonate and proton. That's the good thing, and this leaves the cell on a bicarbonate chloride exchanger, kind of like the ones we had in red blood cells. You may remember those. Don't worry about what happens to the chloride. So far so good. My bicarbonate is back in the body. But what about this? I made a proton. That's acid. And I don't want that coming back into the body because protons are what my problem. I've got too many of them already. I've got acidosis. So how do I make sure that this proton doesn't go back to the body? Probably the best way to do it is to make sure it goes into the urine, down into the tubule. There's a couple ways I can do that. One of them, I have a proton ATPase, an active transport pump which will take protons and shove them into the urine using ATP for the purpose. That's great. That does present a problem long term, though, in that every time I do that, I make the inside of the cell a little more negative and the outside a little more positive, creating that membrane potential. That may limit how long I can do that, or it might result in other things being drawn into the cell that I don't want. So, these cells have another option, which is a proton potassium antiport ATPase. This will allow protons to get pumped out of the cell in exchange for bringing some potassium back in. So this is an active ATPase antiport. Protons out for potassiums in. That is electrically neutral. One positive charge out, one positive charge in, so that can run more easily without producing any ongoing membrane potential. Net result is I started with the CO2 in the blood, converted it to proton and bicarbonate, pushed the bicarbonate to the blood, and the proton out to the urine. This will make my urine more acidic and my blood more alkaline, which is good. That's exactly what I want in this case. All right, so that's the way that our DCT can help us deal with acidosis. It also has a way of helping us deal with alkalosis. Let me quickly show you what that is. So, go. All right, let's talk about how the DCT handling alkalosis. This is going to happen in a DCT type B intercalated cell. So here, if I'm in alkalosis, my body is too alkaline and I need to acidify it. So now my goal is going to be to take a CO2 in here and return a proton. Let's see how it works. If I bring CO2 in, I'm going to combine it with water using carbonic anhydrase to produce proton and bicarbonate. Now my goal now is to get that proton out of here. Well, let's see. What ways do I have to move protons out of this cell? I seem to remember hearing something about a couple of ATPases that could do that. Either a proton ATPase or a proton potassium ATPase. Those may look familiar. And bicarbonate ions could be moved into the urine in exchange for chloride coming in. Yeah, that would work. My end result would be a carbon dioxide converted to proton and bicarbonate. Proton moved back to the blood, making it more acidic, and bicarbonate moved to the urine. This one's relatively easy to remember because it's exactly like a type A cell, just with the transporters and the other membranes. So kind of just flip the cell over almost and use it to combat the other version of the pH condition. Now, how the body decides which of these cells to use and how it turns them on and off, that's beyond our scope right now. But those methods, PCT reabsorbing bicarbonate and the PCT synthesizing bicarbonate and the DCT synthesizing bicarbonate to help with acidosis, and the DCT making protons to help with alkalosis, are major ways that the kidneys can help us handle pH disturbance. All right, and that's the end of the urinary system. Good job. Next, we'll be moving on to the endocrine system. So, see you then.